Have you ever wondered what goes on inside your body? Well, think of your body as a factory full of conveyor belts and machines, all working together to keep everything running smoothly. Countless reactions are taking place inside your body, and each of those reactions take time to occur. Imagine each reaction as a mountain, and you have to climb to reach the other side. The top of the mountain represents the reaction happening, and the other side is the completed product. Now imagine an enzyme coming in. The enzyme would make the mountain much smaller and easier to cross. That's because enzymes speed up reactions by lowering the activation energy, the energy needed to start the reaction. This is an enzyme. Enzymes have a special region called an active site. This is where the substrate binds and the reaction speeds up. Now there are two main models that explain how enzymes work, the lock and key model and the induced fit model. The lock and key design works just like a real lock and key. Think of the lock as the enzyme and the key as the substrate that fits perfectly into it. Now let's look at a real example, an enzyme in your body called carbonic anhydrase. This enzyme helps your body convert carbon dioxide into a form that can travel in your blood and then be released in your lungs. It only accepts carbon dioxide into its active site because the site is shaped precisely for that molecule, like a lock that only one key can open. For example, if oxygen or another gas tries to bind, it would be unable to fit because the enzyme is so highly selective. Now onto the second model, the induced fit model. Think of the enzyme, this time, like a rubber glove. And when a hand slides in, the glove slightly changes shape to fit around it perfectly. An example is hexokinase, an enzyme that uses ATP to add a phosphate to glucose in the very first step of glycolysis. When glucose and ATP bind to hexokinase, the enzyme's shape shifts slightly to fit them better. It then transfers a phosphate group from ATP to glucose and alters the active site, and when released, they become glucose 6-phosphate and ADP, the leftover form of ATP. So what's the difference between these two models? Well, in the lock and key model, the active site is rigid and only one very specific substrate can fit. Think only one key can open one lock. In the induced fit model, for instance, the active site is flexible and can slightly adjust its shape to bind similar substrates, while still remaining highly specific. The biggest takeaway from these two is the lock and key model is very rigid and highly specific, while the induced fit model is flexible. It usually binds to one substrate, but can adapt slightly to others. So which design is more widely accepted in science today? The answer is the induced fit model, because enzymes, while highly specific, are also very flexible and able to adjust their shape to make reactions happen efficiently. The main takeaway is that enzymes are biological catalysts that speed up reactions. The induced fit model remains the most accepted today in science, and enzymes are essential in medicine. They help produce antibiotics, insulin, and vaccines. Without enzymes, our world and our bodies would be quite different, and modern medicine would not be what it is without the power of enzymes.